Hello, everyone. Giving you a bit of time to find your seat. My name is Camille Bouget. I'm the co-founder and CEO of Scientalab, and today I will give you a glimpse of how foundation models are transforming immunology. So I'd like to start this talk with a quick question. Who amongst you has a loved one that is personally touched by a disease of the immune system? Okay, quite a lot, yeah. I'm happy that you're raising your hands because usually when I pitch Scientalab, there is always somebody that tells me they have a loved one that is touched by one of them. Well, this is normal because actually these pathologies are touching 5 to 7% of the Western societies. They are characterized by a dysregulation of the immune system that is too active and is triggering a series of pathologies with heavy symptoms and consequences on the life of patients, usually with a strong effect on their quality of life. What is interesting is that on the first look, they have nothing in common. As you can see, they affect each of one a different part of the body with a different type of symptoms. However, bear with me for a minute and you will understand why it might be more complex than it looks. So to illustrate this, what I'm saying, I'm gonna use the example of anti-TNF. They are the most well-known class of therapeutic agent in this type of pathologies. So if I take the case of rheumatoid arthritis, by example, we see that anti-TNF are working very well for 60 to 70% of the patients. However, in 30 to 40% of the cases, they don't have any effect or a low effect. And this is mostly due to the fact that they have been developed with a one-size-fits-all approach, meaning that we are seeing every patient as the same and developing a drug for this persona. What is interesting to see is that we observe this same effect of 60 to 70% of effect across all these pathologies. So all these drugs have this quite moderate effect. However, if we dig a bit further, we see that this anti-TNF, by example, they also work for 60 to 70% of patients with diseases that don't have anything in common in appearance, like lupus or Crohn's disease, by example. So we start to see here that despite very different looks in appearance, there might be some underlying shared biological pathway that could explain this shared therapeutic effect. This is actually the holy grail of the modern healthcare system, to understand why these patients are the same and to find something that could qualify them easily and bring them into one of these subgroups to offer them a personalized therapeutic approach. I think you all heard about biomarkers. Biomarkers are actually this measure that helps us qualify these patients. Most of these biomarkers have been developed by traditional research approach. This is okay, they work very well, they are used every day in clinical practice. However, with the recent boom in new AI technologies, we think that there is a better way and a faster way to develop them at scale. There is one technology in particular that we are very interested in at Scientalab, and this is the foundation models. So foundation models, unlike the first generation of AI that was automating a task, basically you had a task in mind, you knew what was the outcome and you were automating it. Here we are on the second generation of AI. So basically, we are trying to identify new signals amongst a big quantity of data. So if I go a bit more into the technical details here, as you can see, we have a big issue with data. We need to have a very big quantity of data at the entrance of the model. However, if we find this data, we can train a model with a self-supervised approach that is going to learn a patient representation. So it's like the brain of a baby, basically, that learned what it's like to have rheumatoid arthritis or lupus, by example. So at this stage, we only have a brain. Okay, it's smart, but it's not doing anything. You need to deploy this brain within what we call a downstream task on a small amount of data, sometimes even no data in the case of Gen AI. And this is where you can identify new patterns and work on new research hypotheses. So usually at this point of the talk, people are a bit confused. So I'm going to illustrate that with an example of a downstream task that we developed at Scientalab. So at Scientalab, we have a first version of a foundation model that is dedicated to immunology and inflammation. What we did is that we deployed this model on the pathology, a specific pathology, which is rheumatoid arthritis, we had a cohort of patients across Europe, and we were trying to understand how these patients are responding or not to different drugs for this pathology. So we obtained results for all the major drugs. However, I'm going to focus here on methotrexate, which is the gold standard in this pathology, just because the results were much more stronger than for other drugs. So as you can see, we were starting by a very hybrid cohort, and we were able to identify 4 different subgroups. The first one is characterized by nothing, which is actually good because it proves that the model knows what it doesn't know. So it's not classifying patients randomly into the 3 other ones. The one for which we don't know what's happening, they are in the first group. So group 2 and 3, respectively, are characterized by a low response linked with a low disease activity and a high response linked with high disease activity. The most interesting subgroup here is the group number 4. As you can see, we have a subtype of patients where the disease activity is quite mild and the lymphocyte count is very high. And for this patient specifically, methotrexate has a very decreased therapeutic effect compared to the rest of the patients. So it's very interesting here because we start to see that among the same pathology, we can stratify patients based on biology criteria. I'd like to emphasize also that we used a very preliminary version of our model here, and we were still able to achieve a 7% increase in performances compared to an unpre-trained model. So it gives you an idea of what a proper foundation model can do in the near future. So this is actually what we do at Scientalab. We want to use this scientific knowledge to redefine the way we understand immunoinflammatory pathologies. The goal is to move from a description of the symptoms at a very clinical level to a redefinition of the endotypes that can be cross-pathologic. And by using this information during the drug discovery and development process, we think that we can develop better drugs at a faster rhythm with better therapeutic effects for patients. And the goal is to say goodbye to the one-size-fits-all approach and finally open the path for the AI-powered precision immunology. Thank you very much, and I think we have time for questions. Yes?
It seems that your model is complex, it's a deep learning model, so how do you distinguish, how do you set aside a specific feature for the lymphocyte level?
Sorry.
As the model is supposed to be complex,
yeah.
How come you can identify the covariate, which is the
yeah.
lymphocyte level specifically, in a model which is supposedly a black box?
Yeah, so that's a good point. Haven't had time to talk about it here. Basically, what the model does, the foundation model, it's identifying this subgroup. So it puts every patient within a subgroup, and it identifies the 4 or 5 subgroups. You need an extra layer of technology that is called explainability, in French, interprétabilité, to understand how this subgroup is characterized. So basically, you dig into the neural network to understand how the predictions were made. And this is how you can achieve this type of information. Yes?
Maybe you said in the beginning, I missed it, but what is the input from the patient data perspective that you're using as an input to your foundation?
Yeah. I don't know if you heard the question is what type of input do we use at the entrance of the model? You can use all type of information. For this project that I presented, we were focusing on clinical and biological data just because we wanted to achieve results that had an impact on the clinical practice. And usually physicians, they don't use sequencing data or genomic informations. However, to move to the R&D process, we do need the omics data. So we developed this layer that can work on clinical information, and we are now working on developing the omic brick. The idea is to have the most comprehensive view of a patient possible to make predictions at different levels of information.
We have like 30 seconds left, so I don't know if
There's a quick
like
question.
Or,
no more questions.
If there are more questions.
So we are at, I think, booth 23 or 24, but you're going to find us somewhere around there if you have more questions, and we are going to be here the whole day.